Thank you, Lorene, for that kind introduction, and thank you to you all for coming out and being with us today. It is an unseasonably wet day here in San Francisco, so it's really not this bad all the time. I'm going to give a little bit different talk than I think most of the other speakers. It's designed to be thought-provoking and to get you to think a little bit differently than current paradigms. The paraphrase title is, "Can even a touch of HIV be bad for you?" So some of the other panelists will talk with you today about many of the mechanisms thought to be responsible for elite control of HIV replication. The various host factors that are thought to control virus replication. And some of the strongest evidence, just to summarize briefly, is for T-cell mediated control. And there are probably several aspects of T-cell mediated control that are important. Certainly a stronger proliferative capacity in killing, as Steve will talk about today, is likely an essential feature. Polyfunctionality of T-cell responses may be an important factor. And certainly the genetics studies tell us that having a protective HLA type is probably also important. And T-cell mediated control may also make the virus less strong. If the virus has to escape the T-cell HIV-specific immune response, it may get less fit and be less capable of replicating. And some emerging research seems to suggest this. There's also some evidence that natural killer cells may be involved. Mary Carrington's lab has suggested that this may be the case with some genetic studies. Neutralizing antibodies and other properties of antibody responses could be important, although there's a lot less research to suggest that that's the case. And then there may be protective factors, the co-receptor for HIV, CCR5. People who are elite controllers may actually have favorable genetics, which make their T cells resistant to HIV infection. And that may be another partial explanation. There are likely several other mechanisms. And why do I list all these things here? Why is it important to recognize all this heterogeneity in the mechanisms by which people control HIV replication? And I think the reason is that not all elite controllers are the same. Everybody's very unique, and everyone has their own way to control the virus. And it also turns out that not all people who control the virus are long-term non-progressors. And so if there's one message from the talk today, it's that it's this Venn diagram here that Steve Deeks and Bruce Walker published a few years ago in a review, that while most HIV controllers are long-term non-progressors, some of them do actually progress. And I think we can learn a lot about the biology of HIV. Probably will have implications for vaccine design if we understand why some controllers progress and why some controllers don't. And one of our initial thoughts on this was whether there were ever any negative consequences to the control of virus replication in elite controllers. So can even a touch of HIV be bad for you? And a very simple way of formulating this is, do elite controllers ever get sick from their HIV? And one thing we can do is look at the CD4 counts. And in our study in San Francisco, this is a sample of 58 of our, the first 58 elite controllers in our local cohort. And we see that compared to HIV uninfected individuals in blue, HIV-infected elite controllers in red do have significantly lower T-cell counts. Most of them have normal T-cell counts, but some of them do have significant and measurable levels of T-cell depletion. And about 15% of our cohort actually has had T-cell counts in the range where current treatment guidelines recommend starting antiretroviral therapy. So despite the fact that they have an undetectable viral load in the blood. And 4 of our 58, or 7%, met the clinical definition of AIDS. 3 by virtue of having a persistently low CD4 count below 200, and 1 who had Kaposi sarcoma despite a relatively high T-cell count in the 500-600 range. So why do some elite controllers experience T-cell depletion despite clinically undetectable viral load? And we wondered whether immune activation may play a role. And why would we think that? Well, immune activation and inflammation has been implicated in HIV pathogenesis, and a lot of these insights have come from animal models. And I want to introduce you to the Sudi-Mangabe monkey on the left. Many of you may have seen similar slides before, but the Sudi-Mangabe is the natural host of SIV infection, the simian immunodeficiency virus. This is where we think at least one strain of HIV, HIV-2, came from these monkeys. And it's a natural host of SIV. It's been living with SIV, we think, for centuries, and maybe even thousands of years. They have high viral loads, comparable, if not higher, to what we see in HIV-infected people, yet they don't get sick, and they don't progress to AIDS. Generally they maintain stable CD4 counts, but they rarely, if ever, progress to AIDS. And a key feature of their illness, or the infection in this setting, is that there's low inflammation and low immune activation. But if you take the same virus and you put it in a different monkey, in this case the rhesus macaque, this monkey has comparable levels of virus replication. Maybe a little bit higher, but comparable. But this monkey exhibits severe CD4 depletion and progresses to AIDS very rapidly. And the difference between these two scenarios appears to be immune activation. The rhesus macaque, just like most HIV-infected people, experiences high levels of generalized immune activation in response to this viral infection. So we're not just talking about the T cells and the B cells that are specific for HIV or SIV in this case. We're talking about the vast majority of the T cells in the body, not just those that are HIV specific that are getting activated. And the height, the extent of immune activation seems to be a strong predictor of how rapidly people progress with untreated HIV disease. It also ends up being an important determinant of how many T cells people gain back during viral suppression. And so we wondered whether, among these elites who progress, whether immune activation may be playing a role. Could even a touch of HIV be bad for you? Stimulate even low, very, very low, unmeasurable levels of HIV replication be stimulating lots of inflammation.